Welcome to the basics of biochemistry, everything that we went over in all of the beginning of freshman year biology, and then again in IB1, and then again in IB2, all crammed into hopefully 30 minutes or less. We'll start off by going through a quick review of the vocabulary for the unit. This will probably be new to you. It is not part of the IB curriculum, is to talk about different isomers, although you may have seen this in chemistry. And there's 3 types that you'll need to know. Those are going to be structural, cis-trans, and enantiomers. After that, we'll go on to talking about the different functional groups. And there are 4 main ones that you need to be responsible for. They're going to be the aminos, the carboxyls, the hydroxyls, and the phosphates. And then once we're done with that, we'll start talking about the organic macromolecules. So you should be very familiar with each one of these organic macromolecules. And I will tell you, I'll try to break this down by color so that as you're watching, if you feel really confident about carbohydrates, then you can skip through that section and know when to start and resume when we go into a different discussion. So any topic dealing with carbohydrates, I'll have in this kind of bright green color. Proteins will be in pink, and you can remember that because your muscles are pink. Lipids will be in yellow, and again, we can think about butter being bright yellow because it's a fat. And nucleic acids will be in this purple color. So again, as you're going through, if you want to skip everything on carbohydrates, just go to the section where you start seeing pink writing, etc. So we'll start talking about isomers by describing what they are. All isomers have the same chemical formula, but some kind of different structure. And so to start talking about this, the most obvious is a structural isomer. So structural isomers are just the most broad range where you have the same formula, different configurations. An example of this would be C2H6O. If I were to ask you to draw that, you don't necessarily have to come up with the exact same as everybody else does. So here are 2 examples of C2H6O. On the left, you have CH3, CH2OH, also known as ethanol. That's common alcohol. And on the right, you have a CH3OCH3. That's dimethyl ether. Very different than alcohol. Some similar properties, but it's used in commercial something or other. I don't know, look it up on Wikipedia. So very different. Those are examples of structural isomers. Next, we can take a look at a cis-trans isomer. Now, anytime you have a single bond, everything is free to move around that bond. You can have rotation around the bond. So everything over here might kind of rotate as compared to everything over here rotating in an opposite direction. But once you put in a double bond, things get locked. There's no rotation anymore, meaning that something that is on one side is going to stay on that side. Let's take a look at fats. Those are the classic example of why you need to know about a cis-trans isomer. In fats, you just have these long chains of carbon, and sometimes you get a kink in them in the form of this double bond right here. So this right here makes an unsaturated fatty acid, and you can have the fatty acid chain go in the same direction like this with a couple of hydrogens. That would be a trans meaning across, like the transatlantic railroad. If you're a history buff, I don't know why that came up, but that would be a trans. If you have it on the same side, that would be a cis. So up here would be hydrogens opposite from each other. And then if we kind of scroll down a little bit, here we go. We can continue these chains on. And so that would be our cis isomer. And finally, a fun word that you can say at a party or impress your family is enantiomer. And so like we said, anytime you have a single bond, they can rotate. However, that rotation isn't going to allow them to break bonds and reform in an opposite way. An enantiomer just means a mirror image of each other. And so we remember that our carbons make 4 bonds, and they make this like tetrahedron shape, and that it's in 3 dimensions. So to illustrate this, we use dotted lines to indicate a bond going backwards or against the plane that you're looking at, and a triangle to represent that it's coming out or towards you. So in this, you can see that I've gotten things opposite. Now, if we were to draw in whatever these functional groups were, say an ethanol and hydroxyl and maybe methyl group here, and going backwards. I have no idea what this chemical would be, but anyway, these are enantiomers of each other. They're mirror images. There's no way that you can have one and rotate it in such a way that it somehow magically becomes the other one. They're just locked and they're not going to do that. And we always call one a left-handed version and one a right-handed version. As far as I know, they're completely arbitrary as to which one becomes left and right-handed, but they have very different chemical properties. And biologically, let's think about pharmaceuticals. You can have a drug that in its left-handed form treats a certain disease, where in its right-handed form it's completely useless. You also see this with amino acids. All of them are left-handed. I might be lying about that, but all of them go in the same direction. So there's your overview of isomers. You've got structural, which all isomers are structural. We can break these down further into cis-trans, trans meaning opposite directions, cis meaning on the same side, and finally enantiomers, which are your mirror images. And we can briefly talk about functional groups. These are just groups that are attached to a simple hydrocarbon. So most biological molecules are going to start as some kind of carbon skeleton. What comes off of them, that can be variable, and there's 4 that you need to know. The first that you'll be responsible for is the amine group. If we take a look at what the actual structure is, that looks something like over here, with a nitrogen, 2 hydrogens attached, and then some kind of whatever that wants to be over here. Shorthand, you're either going to see this as NH2 or H2N, depending on which direction you're going. The second is a carboxyl group, which you'll see pretty frequently. Both of these you'll find in amino acids. Your carboxyl group is a C double bonded to an O with an OH group. That OH group is the hydroxyl group. We'll look at that in a second. And then your COOH is your shorthand. It's a much faster and easier way to write it, obviously. So your OH here is a hydroxyl group. You'll frequently see these in alcohols. So alcohols always have an OH attached to them, and anything with an OH you can classify as an alcohol. And the last one here is a phosphate group, which is a PO4 2-. Again, remember that this bond right here can rotate, so you don't have to remember that the double bond goes up. It rotates. However you draw it is fine, so long as you remember to have 2 O negatives. So here's your quick recap. So let's go ahead and get started about each of our organic macromolecules, starting with the conversation about a carbohydrate. Of course, you should remember that each of your macromolecules is made up of smaller monomers. So when I refer to a monomer, you should be thinking about how beads make up a necklace or how bricks make up a house. They're the small pieces that make up something bigger. And in the case of a carbohydrate, we're always going to have our monomer as a monosaccharide. And our general formula for all monosaccharides are C6H12O6. The general formula for a carbohydrate is going to have a ratio of 1 to 2 to 1 in talking about carbons to hydrogens to oxygens. So the function of carbohydrates, the main one, is going to be quick energy. We're talking about 4 calories per gram, so quite a bit of oomph, and this is going to circulate in your blood as glucose anytime you need sugar to be used for cellular respiration. And AP is going to look for you to know that there's 3 different classes of carbohydrates that we're going to look at. Monosaccharides, meaning one monomer of carbohydrate, disaccharides, which have 2 monomers attached to each other, and polysaccharides, which have many. And so we're going to start by looking at the monosaccharides and what you should know about them. There are 3 monosaccharides you need to know: glucose, galactose, and fructose. All of them are isomers of each other, so they all have the same chemical formula and a different structure. And then if we take a look down here at the disaccharides, again, there's going to be 3 different ones that we want to talk about, and how they form together is going to be just as important as understanding what they each are. So we'll start with a brief conversation about how disaccharides form. So what I've shown right here are 2 saccharides next to each other. I've got the rest of the drawing over here that I just didn't really care about drawing out. And here's what's important, is that for each carbon, there's a hydrogen and an OH group or a hydroxyl group attached. And again, on the other side here with this monosaccharide, there's a hydroxyl and an H group attached. So what's happening right here is very important. We're going to have these coming together and being removed. So now a bond becomes formed between these two carbons. That should be a single bond. I just made it really, really thick. And you'll notice what you're left with is water. So this is your dehydration synthesis. You formed something by taking out water. And monosaccharides form into disaccharides, form into polysaccharides, all by doing that dehydration synthesis reaction. And conversely, we can break bonds by talking about a condensation reaction, exactly the opposite, where you have something like a bond which is already formed, and then you reintroduce water into that to break up that bond. Add your OH here, your H here, and now you've got two separate monosaccharides, or in the case of really any other macromolecule, exactly the same thing is happening. And so let's take a look at the 3 different reactions that you need to know about disaccharide formation. So all of them start with one glucose. I've abbreviated that GLU. In the case of maltose, you need a glucose plus a glucose. If you like malted beverages, that's the sugar that's there. Glucose and galactose are going to give you lactose, which you're going to find in milk. If you're lactose intolerant, it's because you're not able to break down lactose into glucose and galactose. And finally, sucrose, table sugar, is just glucose and a fructose. Fructose is when they talk about high fructose corn syrup, that's the fructose that they're talking about. So, and in all of these cases, we're starting with glucose. And again, remember that glucose is going to be what's important for being able to feed the brain, especially the brain, but the rest of the body as well, by doing cellular respiration. And we can zoom back out here and talk about our last topic, which are the polysaccharides. And the polysaccharides are going to have 2 different functions, depending if you're a plant or an animal. They're going to be structural and storage. We'll do a little zoom in here. Plant structure is primarily going to be cellulose. Cellulose makes up the plant walls, or the cell walls of the plant. So cellulose, uber important. By the way, you cannot digest it. If you could, carrots would have some of the highest content of calories that you can imagine, or celery. Luckily, we can't, so there you go. As far as for plants, their energy storage is going to be found in starch. You can break this down too. So next time you go enjoy a potato, just remember you're having a polysaccharide. That'll get broken down into disaccharides, monosaccharides, and into tasty num-nums. In terms of animals, structurally, now we don't have this, but chitin, and that is the exoskeleton of bugs. Also, quite interestingly, it is the structure of the cell wall for fungi, giving credence to the idea that animals and fungi are more related than animals and plants, or fungi and plants. And lastly, in animals, our energy storage is going to come in the form of glycogen. Remember this from our unit talking about homeostasis. So there we go. Quickly recap all of this when we're talking about any of our carbohydrates. Nom nom nom. The monomer is going to be the monosaccharide. Here's your chemical formula. Here's your generic formula, or your reduced formula. Our functions, for the most part, we're going to look at quick energy, but when we come down here and look at polysaccharides, also remember that there's additional functions there. 3 classes that you need to know: monosaccharides, disaccharides, and polysacs. And so for the monosaccharide, you just need to know glucose, galactose, fructose. For disaccharides, you need to make sure you understand how they condense, and as well as what combination they can condense to form. And again, polysaccharides, your plant and animal functions. Now we'll start talking about proteins. We'll start with their monomer, which is the amino acid. Here, very briefly, is again the structure of an amino acid. We see the carboxyl group over here, written 2 different ways. The amino group over here, again written 2 different ways. And up here at the top is your variable group. You have 20 different amino acids. Each one has a different variable group and is coded by a different codon of DNA. We'll get there later. But this R group is going to be very important in the characteristics of the protein. And like we did with our carbohydrates, we need to look at how a dipeptide forms. So I'm just going to look at this part over here. So our carboxyl group and this part, so I've got 2 separate ones going on here. okay, and they're lining up next to each other, we're going to lose the water and form a bond. And then these will kind of just straighten themselves out. And so that is going to make your peptide bond for your dipeptide. So anytime we're talking about a covalent bond in a protein, those are called peptide bonds. And on to functions. Remember, if you don't remember this, Miss Unger hits me. Okay, HITSME, so you can remember it that way. Hormones would include things like insulin. Your immune system needs it for your making antibodies. The transport would be any kind of membrane-bound transport, so we're talking about moving things into and out of cells. For structure, we're thinking the microfilaments and the microtubules that make up the cell structure. In muscle, actin and myosin, and arguably the most important are the different enzymes that are going to be involved in all the biochemical reactions in your body. So now that we know the microstructure of amino acids and how they come together, we're going to look at how an entire protein is formed, starting from the primary structure, and that is simply the sequence of amino acids. One small problem with your sequence of amino acid could have catastrophic effects. We can be thinking about how just switching valine for glutamic acid causes sickle cell anemia. We can think about a lot of different genetic diseases. Switching out one amino acid for another can lead to huge problems in the rest of the folding, and that's because the primary structure is going to lead to the interactions that happen along this strand. So we can think of that primary structure as just a long strand of beads. It's not doing anything important. It's not very exciting, except for dictating how secondary structure is going to come about, as well as how tertiary structure is going to come about. And so in talking about secondary structure, we're thinking about the folding of the primary structure. So we've got these long chains that start to fold back on itself, either in beta pleated sheets or in little helices called alpha helixes or helices. Once you get the secondary structure figured out, and remember these are held together by hydrogen bonds all throughout here, which is keeping everything nice and sturdy, we can start talking about the overall structure. So secondary structure is very localized, tertiary structure is all over the place, right? So we can have a couple beta pleated sheets with some alpha helixes and then just globular crap, and then maybe some more beta pleated sheets, and so on and so forth. So tertiary is going to mean the entire chain of amino acids all folded up. And lastly, sometimes you're going to get a quaternary structure. This is the final quaternary. I need to focus on spelling. This is the final level of structure, and this is when we introduce prosthetic group. And just like in a prosthetic leg, that doesn't necessarily mean it's made out of the same material as the rest of the leg. It still becomes part of the body. And in the case of a protein, we might have something like, the polypeptides with a couple of irons in here. That's iron. And you'd have 4 of these, and that would make hemoglobin. Okay. So there we go. To recap that just a little bit and make sure we're perfectly clear on that. Primary, we're talking about what the different amino acids that go together are. So if we kind of scroll up here a little bit, whatever these R groups are, those are the sequences that we're worried about. This is all going to stay the same. So one of these is going to join on with another one of these using this condensation reaction right over here. So you've got one attached to another, attached to another. That's going to be the same for every protein. Not exciting. This is what matters, right? So then once you've got that primary structure established, the secondary structure comes from there being hydrogen bonding between the R groups of amino acids that might be next to each other. Think about this as a long chain of little kids holding hands. Eventually that chain is going to start folding in, and people who are friends are going to want to be near their friends, and then they're going to just kind of be attracted towards each other and fold the whole group over and over. That's your beta pleated sheet or your alpha helix. The tertiary is the overall structure of an entire strand of amino acids. In this case, this is called the polypeptide. And finally, the quaternary is your final structure of proteins. Now, to go back a second, whoopsies, there we go. To go back a little bit further, as we said, the secondary and then again the tertiary and quaternary structures are all held together by hydrogen bonds. And hydrogen bonds are strong compared to not having a bond, but pretty weak in the world of like bonding. Okay, so we've got these weak bonds, and they're kind of like high school love. I love you guys, but sometimes not the strongest thing ever. And you put a little heat on it, make things uncomfortable, and things go haywire. So in the case of proteins, you add a little bit of heat to it, and you can have the process of denaturation happen, which is where those bonds that were holding the beta pleated sheets together, if those bonds break down, things just get all loosey-goosey. It goes downhill from there, and you start to lose the actual function of what your protein was. And I've drawn you a nice little light bulb that's indicating here's something that you need to know for the AP test that you don't need to know all of them necessarily for the IB. For IB, you need to know that there's hydrogen bonding and that there are disulfide bridges between some of the R groups. AP is going to be a little bit more particular about what they expect you to know. So let's go through that. You're going to have hydrogen bonding. That's the most common, and that's going to be between the R groups. You're also going to have some ionic bonding between the R groups, as well as hydrophobic interactions, which means if we've got some twisting going on here and here, and these two areas are both hydrophobic, and you put them in a water environment, that they're going to attract to each other and be folded in such a way that they're not going to be facing out towards the water. So those are called hydrophobic, water-fearing interactions. Van der Waals attractions, those are caused by the fact that if you've got an atom and you've got the little nucleus here, sometimes the electrons are over here and sometimes they're over here. And so van der Waals attractions are just kind of very, very brief and weak interactions based on the fact that electrons aren't always evenly distributed. And finally, those disulfide bridges. And they're going to call them a disulfide bond, possibly disulfide bridges. But all in all, 5 of them you need to know. One more idea about protein folding. This is going from the primary to the secondary to the tertiary to the quaternary structure. And the fact is, we don't know how it happens exactly. But one of the recent discoveries is of chaperonins. And again, this is something AP is going to want you to know that IB doesn't necessarily need you to. And chaperonins are proteins that assist in the folding of other proteins. We're not entirely sure how they work. But one consequence of this is something called a prion. If you have a misfolded protein that's acting as a chaperone protein, then that prion is going to spread to other proteins, which are then going to become prions themselves. This is where you lead to things like mad cow disease, which isn't actually a disease caused by a pathogen so much as it is a misfolded protein that is telling other proteins to fold incorrectly. We also see this in Alzheimer's and Parkinson's, although it also has genetic links. And lastly, IB doesn't care anything about how you study proteins, but AP does. I believe there's a lab on this. Don't hold me to this, but take a look for that as well. So that last little AP concept here is going to be about studying proteins. And there's 3 main ways that we're going to do it. I'm just going to state them very quickly and then give you a little bit of detail about them, but I'm not going to write everything down. So studying proteins, the first way is going to be X-ray crystallography. This is where you take proteins and get them in a crystallized fashion, shoot X-rays through them, and see where the diffracting patterns go. This was common in the 1950s. It's how Rosalind Franklin was utilized to be able to find the structure of DNA because she had been an expert in X-ray crystallography and then moved on to trying it with DNA. The second way is something called NMR, which is nuclear magnetic resonance. This is taking a sample of protein, putting it into a machine, and it measures how different atoms within the protein interact with each other. This will give you a little chart, and this is going back to my organic chem days, so I might be a little bit off here, but it'll kind of give you peaks, and each peak represents what kind of frequency a particular element is getting exciting at. So if you have a high amount of that element, then that tells you. And the third way is called bioinformatics. Now, this is my personal favorite. It's a growing field, and it's one I'd like to be able to look at more in IB when we look at our evolution unit. But bioinformatics looks at the DNA sequence. We put them in a huge database, and then once you look at the DNA sequence, find out what proteins are coded from them. The informatics part refers to the idea that we're putting all of these sequences in a database so that they can be compared to other proteins, other sequences, and ultimately we can start looking at evolutionary relationships. But that's a little bit of a side note. So to study proteins, we've got X-ray crystallography, NMR, and bioinformatics.